Sir, we are on live, sir. YouTube.
Yeah.
Radha madam, it is over to you.
Ramakrishna sir, will you please?
మాట్లాడతారా సార్తో?
Ramakrishna sir, will
Yes,
you
madam.
please
Yes,
share
madam.
college video?
No, madam. You can directly go for introduction.
Sir, please share for 2 minutes.
Yeah, okay. Only 5 minutes.
Very good morning. Myself, Dr. Kumar V.S. Namani, Director, Sri Vishnu College of Pharmacy. It gives me great pleasure in inviting each one of you for session 2 of day 3. And today's speaker is Dr. K.V. Ravanmurthy Garu, and is my teacher, mentor, guru, and whatnot. So the talk sir has selected is nanodegradable systems, current trend and future perspectives. I'm sure this talk will be enlightening to all of you. Sir has several decades of experience, guided several PG students and few tons of PhDs. And this talk is, I'm sure, very relevant and you all love it. With this, I request Radha Madhavi to formally introduce today's speaker.
Good morning to all the dignitaries. I am Dr. Radha Madhavi, working as Associate Professor in Sri Vishnu College of Pharmacy. I feel immensely delighted to present the profile of Professor K.V. Ramanmurthy sir, retired Principal, AU College of Pharmaceutical Sciences, Andhra University, Visakhapatnam. Professor K.V. Ramanmurthy sir had completed his B.Pharm, M.Pharm and PhD from Andhra University, Visakhapatnam. Sir served as lecturer, associate professor, senior professor and principal of AU College of Pharmaceutical Sciences, Visakhapatnam. At present, sir is serving as a special officer, Center for Advanced Scientific Research, RUSA 2, Andhra University, Visakhapatnam. Professor K.V. Ramanmurthy sir held many professional positions like Chairman, BOS, AU College of Pharmaceutical Sciences, Member, Consent for Operation, AP Pollution Control Board and Member, Academic Coordinator, RUSA, Andhra University, Member, NAC Steering Committee, Andhra University, Placement and Training Officer, at University College of Pharmaceutical Sciences, Andhra University. Sir has guided 64 PhD students and more than 140 MCOM students. He has contributed enormously to the research field by publishing more than 150 research papers and presented almost 100 research papers at various conferences. Professor K.V. Ramanamurthy sir has completed 12 research projects and has got 4 patents to his credit in the field of pharmaceutics. Sir was awarded with many awards such as Dr. LVG Naggun's Professor C.G. Shishu Best Researcher Award in Pharmacy by APTI, Srishti Gandhian Young Technological Innovation Award in 2019, State Best Teacher Award in Pharmacy by Government of Andhra Pradesh, Dr. Sarvepalli Radhakrishnan Best Academician of the Year Award by Andhra University. And in addition to this, Sir was awarded with many travel grants. With this brief profile, I am honored to invite Professor K.V. Ramanamurthy sir to deliver the presentation.
Thank you, Madhavi, for your nice introduction about me. You have spoken very good words.
Thank you, sir.
Yeah, I think I'll. I think, is it visible to everybody?
Yes, sir.
Yes, sir.
Can you go into the presentation mode, sir?
Yeah, I'll move it. Now I think it is the presentation mode. Is it okay to everybody?
Yes, sir.
Yeah. A very good morning to all the participants. Director Kumar, Principal Professor Nataraj, faculty members of Vishnu College of Pharmacy. A very good morning to all of you. Today, I would like to present the topic on nano drug delivery systems, current trends and future perspectives. And I'm going to cover about the introduction about the nano drug delivery systems. What are the challenges in their development? What are the regulatory considerations that are to be followed by the Indian scientists for development of a nano drug delivery system into a commercial formulation? And what are the future perspectives that are contemplated for these nano drug delivery systems? Why people are looking at nano drug delivery systems nowadays compared to the conventional drug delivery systems which are well developed? The reason being, the conventional drug delivery systems, when they are administered into the body, they are being absorbed, moved into the systemic circulation, subjected for various processes, and they will be exerting their pharmacological effect in general. And in this particular course of travel of these conventional dosage forms, they are going to touch different parts of the body, exerting their action on the body, as well as these are being subjected for different actions by the body particles. And because of that, we may be facing so many side effects besides the beneficial therapeutic effect. So when we design a nano drug delivery system in a suitable way, such that it can be tailored in such a way such that it will be reaching to the targeted site in the body in a predetermined rate without disturbing the other parts of the body. And after reaching to that particular targeted site, they are going to exert their therapeutic response with minimal side effects. That is the reason why many of the scientists are now aiming for developing the nanopharmaceuticals. And these nanopharmaceuticals may constitute from physical side, chemical side, or biological side. And if you look at the list, US FDA has approved about 100 nanomedicines for the commercialization, and they are well in the US market and other markets. So what is a nanoparticle or a nanopharmaceutical? According to the European Commission recommendation, nanomaterial refers to a natural or incidental, that means accidentally or created or manufactured willfully, which is comprising the particles, which is either in an unbound state or in an aggregate state, where one or more external dimensions is in the size range of 1 to 100 nanometers. So here we are considering not all the 3 dimensions, but at least one dimension. It should have a dimension in the range of 1 to 100 nanometers, and the number of particles that are being counted should be more than 50% of the total mass that is considered, and according to the number size distribution, not the weight size or other distributions. So according to this European Commission, all the particles which are having the size range into 1 to 100 are considered as nanomaterials. Why these nanostructures are important and advantageous for us? So if you just compare this particular diagram, where I have given the different size ranges, the human cell is having a size range of approximately 10 micrometers, and bacteria is having a size range of approximately 1 micrometer, and the antibodies and viruses are having the size range between 10 to 100 nanometers, and our nanopharmaceuticals are going to comprise in the same size range. So these can be well targeted to the viruses and antibodies in the body very efficiently. And below that size range, we have the proteins or DNA molecules or the water molecules. So the surface area of the nanostructures is extremely large relative to its volume. That is one of the best advantage. And if you just take an example of a silicon nanowire, which is having a 10 nanometer in the diameter, it has only 40 atoms in it. But based on this assumption, if we take a cross section of the square, then it can approximately have 1 out of every 10 atoms which are going to be associated with the surface of the nanowire. So every 1 atom is going to associate with the surface of the nanowire. And if you take the magnified part of the same wire, which is having a 10 mm length, width of the 10 mm, only 1 out of 10,000 atoms are going to be associated with the surface of this wire. So by increasing this size by 1,000 times, we are going to have 1 in 10,000, whereas by decreasing the size by 1,000 times, 1 out of every 10 atoms are going to associate with the surface. Because of this important character, these nanostructures are going to be very, very important, because these nanostructures are going to associate with the surface of the different biological membranes, and that's why they are being considered as very important in the formulation development. And I'm just giving a broad classification of these nanostructures. There are many other classifications. Let us not worry for that, because it becomes another big topic of discussion. And hence I comprised myself for only few nanostructures in a broad way, and they can be divided into nanocrystals, polymeric nanoparticles, liposomes, micellae, dendrimers, mesoporous silica nanoparticles, inorganic nanoparticles, and carbon nanotubes. I'm going to just give the brief characteristics of these different nanostructures. And starting with nanocrystals, these are carrier-free drug particles. That means we are going to use only the drug particles which are being manufactured as nanocrystals without any other carriers involved in that. So these are crystalline particles produced by different methods like parallel milling, high pressure homogenization, precipitation process, etc. And we have the option of modifying the surface characteristics of this particular crystalline nanocrystals, and based on the surface modification, we can achieve prolonged targeted release with low doses and decreased side effects. And the major advantages of these nanocrystals are improved surface area and improved dissolution rate of drug, and it can improve the saturation solubility of the drug, and it is very safe composition because it is not containing any other carriers, and it is suitable for intravenous injections, and it can create a very high drug bioavailability. And these are the different marketed nanocrystal formulations approved by European Union and US FDA, and they are well in the market, serving the mankind to the great extent. And if you look at these applications, majority are the anti-cancer applications, or for immunosuppressant for kidney transplants, etc. And the next part is polymeric nanoparticles. These are prepared as nanospheres or nanocapsules by using methods like nanoprecipitation, double emulsification, polymer coating, and emulsion diffusion. And they can be applied for various applications like gene delivery. They can be used for crossing the blood-brain barrier or improving the oral bioavailability, or they can be used for cancer targeting. And they can be administered by infusion, injection, or oral ingestion. And the characters can be tuned according to the requirement, and they can carry different multifunctional agents. And they can have very good thermodynamic stability, and they can penetrate deep into the cells and tissues to a great extent. And these are the different polymeric nano formulations that are being marketed. Again, majority are for tumor or something like that. And next comes the important nano formulation, liposomes. And we all know a lot about the liposomes, hence I don't want to discuss much about this. And these are synthetic vesicles formed from lipid bilayers, which may be divided into two major groups like unilamellar or multilamellar, and they can dissolve both water soluble and lipid soluble drugs at the same time. And they can be used for passive targeting or they can be used for active targeting. The highly efficient cargo delivery and reduced cargo toxicity. So these are the major advantages, and they can have different applications like conventional liposomes or pegylated liposomes for drug targeting, or ligand targeted liposomes, or we can create a multifunctional liposomes for thernostic applications. And these are the different marketed liposomal formulations available in the market. And micellase. And these are the spherical amphiphilic copolymer nanoparticles which are formed by supermolecular assembly and having a core shell structure with hydrophobic interior which is separated from the aqueous exterior. So the major advantage is they can be protected from the non-specific uptake by the reticuloendothelial system because of their hydrophilic surface characteristics. And they have very high drug loading capacity, very good stability in the blood, and they can have prolonged circulation times because of their hydrophilic nature and less number of side effects, and they can protect the drugs which are hydrophobic in nature in the inside core of the micellae such that the drug cannot be degraded in the body. And if you look at the commercialization of these micellae, they are only under the development stage, and they have prepared the doxorubicin micellae which are under the preclinical stage, and they prepared the paclitaxel micellae which are under the phase 3 clinical trials. And another important nanoparticle is the dendrimers. So these are synthetic tree-shaped macromolecules having a 3D monodisperse structure, and they're having different branches extended from the central molecule, and we can have a predictable size according to its generation number, and the size range of these dendrimers will be in the range of 1 to 5 nanometers. So they are having the free end groups, and they can be easily modified for conjugation, and they are biocompatible materials, and they can reduce the cytotoxicity, and they can increase the permeability of the drugs across the biological membranes. And the specialty of these dendrimers is they have the defined molecular weight, they are having a uniform shape, and they can have the host-guest entrapment properties where drug molecules can be entrapped in the central core of the particular dendrimer molecule like this. And we can have different functionalization characteristics to the surface groups that are provided here, like the pegylation or targeting properties, etc. And the other best advantage of these dendrimers compared to other nanoparticles is the extremely low polydispersity index, and hence they can be have very uniform size range. And the dendrimers under the development stage are drug and acetylcysteine under the phase 1 clinical trials. And mesoporous silica nanoparticles. The mesoporous form of silica particles in nanoscale sizes with a large surface area and solid structure. And they have very high loading capacity, and they have good protection ability by keeping the carbo molecule inside the pores of the silica nanoparticles, and they are used for controlled drug release ability. And they are also under the development stage, and they are under the different stages of studies like preclinical. Almost all of them are in the preclinical stage of the development. And inorganic nanoparticles, again, they are having very good applications in the other fields, and their medical applications are now being explored to a great extent. And various metallic elements like silver, gold, copper, zinc, magnesium, etc. are being used for the preparation of these inorganic nanoparticles. And the major advantages of these inorganic nanoparticles is They can be customizable for the size. They have large volume to surface to volume ratio, and they have very good optical properties and surface plasma and resonance properties. And method of manufacture or synthesis is very easy, and we can do a lot of surface functionalizations for these nanoparticles, and are very less toxic to due to their inert nature. And there are many nanoparticles that are reported for their therapeutic applications. And again, there are also majority of development like doxorubicin and aptamer. And we have the next carbon nanotubes. They are needle-like carriers. They can easily by and they are categorized as walled. Amarao. I think somebody is causing disturbance, please.
Yeah, he is muted, sir. Please carry on.
Please. So they are categorized as single walled, which have high near infrared optical absorbance, or multi-walled, having unique physical properties like Young's modulus and electrical conductivity. So they have very high elastic modulus and mechanical strength, and they have high electrical and thermal conductivity. They have very high prolonged circulation time, and they can have good cell membrane permeability and high aspect ratio, which will allow for high drug loading. And they are also under the preclinical stage of the development for the drug epidermal growth factor and cisplatin. Why these nanoparticles are important? As I told you in the beginning of my talk, the major goal of the preparation of these nano drug delivery systems is for reducing the side effects and for improving the therapeutic indices. And they are well studied in the cancer therapy. For example, a product called as Doxel, which was prepared by liposomal formulation of containing phospholipids, cholesterol, and PEG, and it is containing the doxorubicin, and it gave very good extended circulation time and elimination of elimination of RES uptake. And Markvibo, another liposomal formulation of vincristine, and it improved its circulation time, and its dose was reduced compared to the standard drug. And lipoplatin is another drug which is a lipid-based formulation under the clinical investigation. It containing cisplatin with different types of phospholipids with pegylation, and it showed very less nephrotoxicity compared to the parent drug. And it is used in the treatment of pancreatic adenocarcinoma, breast cancer, and advanced gastric cancer. And European Medical Agency recognized this as an orphan drug because of its success rate for these rare diseases. And another drug, another formulation called as Cardkyla, which is a drug antibody combination, and it was made as a combination of mitonsin derivative with transditimab, and it is used for the treatment of breast cancer. And Abraxane, it is an albumin conjugated nanoparticle paclitaxel for improved therapeutic response. And Rapamune is another type of micellar nanoparticle drug delivery system containing the rapamycin. So all these trials are very successful in improving the therapeutic indices as well as in reducing the side effects in the cancer therapy. And applications in infectious disease was also well tried for these nano formulations. And whenever we take any antibiotic, the frequency of development of resistance, increasing the side effects due to increasing the dose, and majority of these pathogens are localized in the intracellular organisms, and hence drug may not reach to the intracellular level to some extent. And development of these nano drug delivery systems may be useful in addressing these issues for reducing the toxicity, for reducing the resistance, and localization of the drug at the intracellular level. And a drug was developed with the brand name Lipoquin, which is a liposomal formulation of ciprofloxacin, inhaled formulation, and it is able to extend the drug release over a period of 24 hours for treating the lung infections with very much reduced side effects and resistance to the organism. And Ambisome, which is an amphotericin B liposomal formulation used for reducing the side effects for treating HIV and disseminated histoplasmosis. And they have developed various vaccines, virosomal vaccines like Inflexal V for hepatitis, Epaxal for influenza, and silver nanoparticles that are developed for eliminating the biofilm which is going to be formed in the medical devices. And the nanoparticles or nano drug delivery systems were also tried in other diseases like cardiovascular diseases, neurodegenerative diseases, etc. But their success rate in their commercialization is not fully explored, and majority are under in the development of the in the development stage or in the preclinical development stage, etc. For example, in the treatment of cardiovascular diseases, scientists have developed the carvedilol liposomes coated with chitosan, and in the neurodegenerative diseases, they developed polymeric nanoparticles, non-polymeric quantum dots, lipid-based nanoparticles for various drugs for treating the Alzheimer's disease. And similarly, for treating the Parkinson's disease also, they have developed the different types of drugs like ropinirole or bromocriptine, etc. So from our group, I can proudly say we have started this work way back in 2010-11 onwards, and we could produce some good number of papers which are published in highly reputed journals like Nanomedicine, etc. And initially we synthesized a thylated dendromer, and it was characterized for the delivery of acyclovir using it as a model drug. And we have also developed the liposomal formulation for cutaneous delivery, and we have developed some other anti-cancer drugs for the Feistin for targeting the brain and breast cancer. And we have also prepared the nanoparticles, metallic nanoparticles, which are synthesized by using the green chemistry synthesis by using ananas squamosa, etc. So these are all our contributions from the laboratory with the help of my good research scholars who worked with me, and we are able to publish these number of papers. Why we have to consider the research in the nanomedicine is very important and critical. So every research laboratory, either in the academic or industrial research, are aiming for development of these nanopharmaceuticals. But there are good advances that are being reported also. But the success is going to be very difficult because the development is going to take a lot of time. Characterization is going to take a lot of time. And even the regulatory agencies are keen to approve these drugs at a faster rate for their benefits. But the success rate as well as failure rate is also equal. For example, the FDA has approved drugs like Doxin, Abroxane, etc., which are in the commercial market. But one group have developed an anti-cancer nanomedicine made with docetaxel, which was coated with a polymer and targeted with a ligand, and it was very successful in the small clinical trials. But when it is subjected for a larger clinical trials, when the size of the clinical trials is increased, it gave the negative results, and hence the total research was stopped. So unless we reach to the level of that particular large size clinical trials, the success rate cannot be predicted with this nanopharmaceutical research. So there is a big gap between the laboratory products to the commercialization, and in spite of having so many advancement in the formulations development, and this may be a diminishing attribute that might disappoint the researchers and industries. So what are the challenges we face during the formulation development of these nanopharmaceuticals? The first one is the physicochemical characterization. Once we are successful with this, we have to characterize for biocompatibility and toxicology. Then next stage is pharmacokinetics and pharmacodynamic characterization. And once all these three steps are completed, we have to develop a process control and scale up. So we have 4 different challenges, starting with laboratory to the industrial application. So how do we do the physicochemical characterization? So the starting point to the characterization is the determination of particle size, then followed by shape and size distribution. What are the aggregation and agglomeration characteristics? What is the crystal structure? What is the specific surface area, porosity, chemical composition, surface chemistry, the charge, photocatalytic activity, zeta potential, water solubility, dissolution rate and kinetics, and dustiness of these nanopharmaceuticals. And there are different techniques that are available for characterization of these parameters, and they can be grouped into different categories like involving counting, ensemble, separation, and integral methods. Just I'm trying to compare all these things in 4 different major groups. So what are the different counting methods that we can adopt? So we can use the TEM, high resolution TEM, scanning electron microscopy, cryo-SEM, atomic force microscopy, and particle tracking analysis, and some other methods. So the choice of the method is going to be very, very important depending upon the shape and dimensions of that particular nanoparticle. So if you are interested in having the 3-dimensional measurement, we have to use a correct method. It may be having a 2-dimensional measurement, because as per the European agency, the size may be in any direction less than 100 nanometers. So we have to characterize it in such a way such that whether it is a film or a tube, etc., proper size is to be proper method is to be selected for determination of the particle size. And major problem what we encounter during the size measurement is the application of high vacuum, and there is a chance of dehydration of these nanoparticles during the size measurement. So one should take proper care in using the right method and right conditions. And the solution for this prevention of high vacuum and dehydration is cryo-SEM, which may be having the prevention of this dehydration under the high vacuum conditions. And ensemble methods, which are used for intensity weighted particle size rather than number particle size. And we may be using a dynamic light scattering method, or small angle X-ray scattering method, or X-ray diffraction methods. So DLS and quasi-elastic light scattering techniques are based on the Brownian motion of the samples, based on which we are going to determine the weighted particle size. And XRD is a very useful tool for obtaining the information about the chemical composition, the crystal structure, and physical properties of the nano drug delivery systems. And the nanoparticles that are prepared must be properly separated into a monodisperse fraction, and we have to differentiate the monodisperse fraction with that of the polydisperse fraction. And the techniques that are used are field flow fractionation, analytical centrifugation, and differential electrical mobility analysis that are used. And the integral methods comprise, we are going to measure the integral property of the particle, and we are going to use BET, the method, adsorption method, the adsorption isotherm, what we have studied during our basic BPharm classes, BET adsorption isotherm, which is a very useful tool. And we are going to measure the adsorption of this inert gas on the surface of the nanomaterial, and based on that, we can measure these integral properties of these particular nanoparticles. And electrophoretic light scattering is used for determining the zeta potential, which is very useful in determination of the stabilization of these nanoparticles in a particular medium. And the next part of the study is, after the physicochemical characterization, we have to evaluate the biocompatibility and nanotoxicology. If we are not going to establish this biocompatibility properly, it may be leading to the toxicological properties rather than considering the advantageous properties of these nanomaterials. So we have to have proper studies to address these particular penetration of these nanomaterials into the cells and tissues, and how they are getting distributed in the biological systems, how they are getting degraded in the biological system, and how they are subjected for excretion out of the body should be well established. And normally in the nanoparticle preparations, we may be using small doses compared to the normal human doses. And once it is being applied to a biological system, we should address this problem properly. Otherwise, there may be improper relationship between the doses and the therapeutic response what are contemplated in the due course of time. And preclinical assessment is also very important. We have to have a proper biocompatibility testing program, which comprises both the in vivo studies and proper in vitro assays for proving its safety. So Keck and Muller proposed a nanotoxicological classification system like our BCS classification system, and based on the two parameters, namely the size and persistency, they are being divided into 4 major classes like BCS class 1, class 2, class 3, class 4. The nanomaterials are also being divided into 4 major classes. Size above 100 nanometers, which are biodegradable in nature, are coming under class 1 because no endocytosis of these particles will occur if the particle size is more than 100 nanometers, and they are considered to be very safe. And size above 100 nanometers, which are non-biodegradable, are coming under the class 2. And class 3, size below 100 nanometers, which are biodegradable, but they are being subjected for endocytosis, and hence they may be considered partially safe, partially unsafe. And size below 100, which are non-biodegradable, are considered to be dangerous for the characterization of biocompatibility. And evaluation of the pharmacokinetics and pharmacodynamics. So we have to evaluate what is the suitability of this particular system for tumor susceptibility or the targeting, how it is getting delivered to the biological system, and what is the therapeutic margin that it is going to have, what is the dosing schedule that is going to be planned or dosage regimen, and what should be the target population, and what should be the selection criteria of this target population. All these are to be addressed properly. And majority of the researchers aimed for the development of these nanoparticles for tumor targeting or brain targeting. And hence, the discussions that are presented in the research articles or cited are majorly concentrated about the tumor targeting. So we have to evaluate the surface physicochemical characteristics, shape and size, and the particle size less than 10 nanometers, it will be preferentially bound to the blood, liver, spleen, kidney, testes, thymus, heart, lung, and brain. So whatnot, majority of the body organs, the nanoparticles can reach, and they can be found in that particular organ. When the particle size is bigger, they may be detected only in the spleen, liver, and blood. So the surface of the nanoparticles also impact their distribution in these organs, because the serum proteins may be having some affinity for binding to these particular nanoparticles, and they may be influencing the cellular uptake of these nanoparticles. So once the 3 stages are completed in the evaluation of these nanoparticles, the last challenge one is going to face is the scale up and reproducibility of the method. Nanomedicine manufacturing processes classically comprises top-down or bottom-up approaches. That means we are going to reduce the particle size to the required particle size, or we are going to create the nanoparticles right from the atomic scale. So top-down or bottom-up approaches are normally followed in the manufacture of these nanopharmaceuticals. So there may be multiple steps involving steps like homogenization, sonication, milling, emulsification, and use of organic solvents and evaporation of these organic solvents over a period of time. And hence, the industrial scale of production, we may be using higher shear motors or higher temperatures or higher volumes of these organic solvents, which may pose some problems during their manufacture. So whenever we are making all these things at a nano scale or in the small scale in the laboratory, we may be preparing a batch of not more than 100 or 200 ml volumes of the system in which the nanoparticles are being prepared. And hence controlling these parameters is very easy. But when once it is going for the industrial production, we have to take care of all these parameters. And any slight variation in the process can change the shape of the particle, or size of the particle, or surface characteristics of the particle, and physicochemical characteristics of the particle. And we should not compromise for the quality and safety of the nanomedicines, and even the therapeutic outcomes should not be compromised in the name of manufacturing under the industrial scale. So we have various approaches as enunciated by the drug regulatory agencies like FDA, European agencies, or Indian drug manufacturing, CDSCO, etc. So FDA is supporting the use of quality by design and PAT technologies, which is an approach recognized for the systematic evaluation of control of nanomedicine manufacturers. So according to the FDA, PAT is a system for designing, analyzing, and controlling manufacturing through timely measurements. That is, during the processing of critical quality performance attributes, both raw materials as well as in-process materials, as well as the processes. What is the goal of this implementation of this PAT is for ensuring final product quality. So PAT is going to be used for evaluating the critical quality and performance attributes and fixing these critical quality and performance attributes to the suitable way such that we can produce a quality product and assure the quality of the manufactured product. And we have to consider the ethical issues involved in the manufacture of these nanomedicines. So majority of the clinical trials that are performed using these nanomedicines are done on the people who are suffering from incurable diseases like cancers, or on the patients who have received many different treatments without any beneficial effect on their disease. So we have to take this as an important point in doing the clinical trials, such that it should not cause any pain to the volunteers who are involved in the trial, because they are already suffering with the pain. And in spite of having very good experience in the clinical trials, most of these participants are unlikely to get any individual benefit by participating in such clinical trials. That is another important point one should not ignore. And whenever we are going for an early stage of the clinical trial, the benefit to harm ratio on health effects caused by the nano formulations are not fully known. And hence, we should be very careful in taking the observations while the initial stages of the clinical trials are being started by the investigator. And last but not the least, very, very important is exaggerated caution exerted by the ethical committees. Whenever we are saying that it is a nanomedicine meant for treating the cancer, I want to go for a clinical trial. So the investigators are asked very, very important questions like, what is the risk involved in the study? What is the benefit that the patient is going to get? What is the likely outcome of the trial? And what are the humanitarian issues that are involved in the particular study? May be asked very importantly by these ethical committees, and the investigator should be able to answer all these questions to the satisfaction. But majority of the investigators are not very clear, because when we are starting a human trial, it is not possible to predict what is going to happen while we are ingesting a 10 nanometers, the nanomedicine, how it is going to move in the body, where it is going to be concentrated, etc., may not be known fully, and it is not that much easy to predict its behavior in the human clinical trials. So there is a gap between the ethical committees and the human investigators who are going to perform these clinical trials, and the gap should be properly bridged by the regulatory and other agencies to bring down these doubts to a certain satisfactory extent. So whenever we go for the regulatory aspects of these nanomedicines, before obtaining the regulatory approval, we have to prove the biosafety of that particular nanomedicine, what is the efficacy of that particular nanomedicine, how it is stable during the shelf life of that particular product, and what is the dose that is being administered, and how long this nanomedicine is to be exposed to the patient for getting the required therapeutic benefit. So there are different agencies in the different countries. In US, there are 5 different agencies like CDER, CDRH, CBER, NTIG, and Nanotechnology Task Force. So these 5 agencies are going to validate the nanomedicine that is being implemented or introduced into the commercial practice, and they ask a lot of data before it is being approved for the commercial applications. And in European Union, we have Committee for Medicinal Products for Human Use, Innovation Task Force, New and Emerging Technologies Working Group. So these are the 3 agencies that are responsible for allowing the nanomedicine into the commercial practice. And in India, we have Ministry of Health and Family Welfare, Central Drug Standard Control Organization, Indian Pharmacopoeial Commission, and National Pharmaceutical Pricing Authority. These are the 4 agencies which are going to play their vital role in deciding the introduction of nanomedicine into the commercial practice. So India, after recognizing the significance of these nanopharmaceuticals, earlier to this particular guidelines, they used to follow the European Union or US FDA or the Drugs and Cosmetics Act 1948 and 1940 and Rules 1945 for the implementation of even nanomedicine into the commercial practice. But looking at the complexity of these nanopharmaceuticals, the government of India has notified the guidelines for evaluation of nanopharmaceuticals in India in October 2019 in coordination with CDSCO, DBT, and ICMR. So as per these guidelines, we have to conduct the safety studies as per the general guidelines specified in 2nd schedule of the New Drugs and Clinical Trials Rules 2019. Earlier, as per the 2nd schedule of the Drugs and Cosmetics Act 1940. But as per these new rules, it is 2nd schedule of the New Drugs and Clinical Trials Rules 2019. And the most important notification is the nano size range should be declared in the product specification, and the particles should be within the claimed nano size range in all given testing conditions during the claimed stability period and final product. That means whenever this product is tested for its particle size, there should not be any deviation which is given on the label specification. So, which is a very important modification that was made by the government of India for assuring the quality of these nanopharmaceuticals. So, according to these notifications or guidelines, the nanopharmaceuticals are categorized into 4 categories. And category 1, here the drug is new and the carrier is also new, and hence we have to establish the quality, safety, and efficacy as specified in the 2nd schedule of the New Drugs and Clinical Trials Rules 2019. That is a very, very lengthy procedure, and it takes more time for the innovator to get the permissions for its marketing. And category 2, here the drug is new, but the carrier is approved as nanopharmaceutical. That means the carrier is already validated by the CDSCO, and hence we have to prove the quality of the drug as a nanopharmaceuticals. So independent studies should be conducted for assessment of the safety and performance of such carrier may not be required, but we have to prove the performance of the drug as a nanopharmaceutical. That should be established, but we need not prove the efficiency of the carrier for its safety or performance. And category 3, here the drug is approved, nano carrier system is new. So we have to give the evidence of safety and efficacy of the nanomaterial, and the final product should be nanomaterial and the final product. We have to prove the evidence of safety and efficacy of both the nanomaterial and the final product should be proved. Then only it will be approved. And category 4, drug and carrier approved in other countries, but not in India. So we have to give the abbreviated or bridging studies as per the 2nd schedule of the New Drugs and Clinical Trials Rules in 2019. So what a research scientist must address specifically for justification of the nanopharmaceuticals. So whenever a research scientist is claiming the advantages or justification of development of a nanopharmaceutical, the justification must be claimed based on the improved safety, efficacy, and reduction in the toxicity profile. And we have to give the reasons for reduction in the dose and frequency of administration, and improved patient compliance, and cost benefit or any other benefit over conventional or traditional drug. Simply as there are no nanoparticles or nanoformulations for this particular drug, I'm going to prepare this is not a valid claim what is acceptable as per the new rules. Any issue arising out of significantly different pharmacokinetics and or pharmacodynamics than that of conventional traditional drug must be properly substantiated. And for suppressing this specific adverse effect or property, a nanoformulation may be prepared, and we can compare it with the conventional or traditional drug in the central different parts of the body like CNS, cardiovascular system, etc., or ophthalmic or immune system. We have to prove that it is very safe, and its teratogenetic potential should also be addressed whenever it is applicable in making a nanopharmaceuticals. So whenever a nanopharmaceutical is being subjected for the approval, the data that should be provided is, it should give the introduction, chemical and pharmaceutical information, animal pharmacology, animal toxicology, human clinical pharmacology phase 1 and phase 2 and phase 3 trials, and special studies, if any, that are recommended, and regulatory status of this nanopharmaceutical in other countries, and samples and testing protocols, and what is the labeling information. So the manufacturer who is applying for this approval of nanopharmaceutical must submit this entire information for the CDSCO for its approval. So what are the future prospects we contemplate with these nanopharmaceuticals? The nanopharmaceuticals can sense the minute differences at the level of single molecules or single cells. There is a major advantage compared to the conventional doses. And it can be having exit sensitivity, and it can characterize the single cell heterogeneity at extremely high throughput characteristics, and it can differentiate the distinct hierarchies and subpopulations. Whenever we are going to use a bulk drugs, it is going to be an average information that we are going to produce rather than the specific information, and majority of the activities that are related to the bulk assays of the drugs are going to be pooled information from the large numbers. Whereas in nanotechnology, it can be a minute information up to the cell level we can generate. So the nanopharmaceuticals can perturb the cells for treating the patients, and they can be designed precisely for delivering a therapeutic molecule to the targeted location based on the individual requirements, for overcoming the biological barriers and altering the inherent pharmacokinetics and pharmacodynamics and biodistributions of the drug. And it also avoids the complications associated with the conventional delivery using the different types of drug like soluble drugs or insoluble drugs, which may be causing the unwanted side effects. So if this nanotechnology is properly envisaged and developed, we can have very good synergistic effect leading towards the personalized medicines. So by integrating this nanomedicine into a single dosage form, we can use it for diagnosis of the patient's problem. We can measure the cells that are affected with this particular problem. We can perturb those cells for causing the therapeutic effect, and it can be treated in the same way. So by using a single nano application, all these 4 goals can be contemplated in future, and it may be leading to development of the personalized medicine. But it will take a lot of time, and it all depends on the how we are going to biomimic the situations, how it is going to be adapted, how it is going to be self-organized, and how it is going to be amplified for achieving this particular goal. And to conclude, the researchers must carefully consider the advantages and disadvantages of the systems that are going to be targeted for the specific site of application. if you just remember, while giving the examples of the nanomedicine that was developed for the cardiovascular diseases, people have developed the liposomes of the carvedilol for overcoming its solubility and dose-related problems. Though the trial is very successful, here, majority of the cardiovascular diseases are not specific to a particular point because we have different types of hypertension caused by different causes, and hence it is very difficult to design a proper nanopharmaceutical formulation for addressing this particular issue. Whereas if it is a cancer, we all know that it is at a specific site with a particular concept, and hence we can easily address this nanopharmaceutical and it can be easily targeted. So one should have a proper understanding of the problem before development of a nanomedicine, and we have to consider what are the advantages and disadvantages of the system that is being developed. We have to use proper evaluation techniques during the development and characterization. A wrong choice may lead to a wrong conclusion. So the scientists must use the proper characterization techniques, and majority of the techniques that are contemplated or explained earlier are very costly, and establishment of such a nanopharmaceutical laboratory may not be that much easy for all laboratories to start. And establishment of scale-up and optimization for reproducibility is very, very essential, which may be another difficult task one a research scientist is going to face. During the in vivo studies, we have to properly address the ethical issues. And last but not the least, we have to adhere to the regulations for their production and marketing. Unless these are properly addressed, the nanopharmaceuticals that are developed in a laboratory scale may not reach to the commercialization stage. And all this is possible not only by a pharmaceutical scientist. So for characterization of these nanopharmaceuticals, for development of these nanopharmaceuticals, we should have a team of scientists, not a single person, comprising experts from pharmacy, experts from chemistry, experts from physics, experts from biomedical engineering, medicine, and a team can give more valuable development compared to a single person. And that may result in the successful development of the nanopharmaceuticals from bench to the market. So I thank the organizers of this particular seminar for giving me this excellent opportunity to share my thoughts on this platform to all the delegates and other members. And I thank the management of Vishnu College of Pharmacy once again for giving me this opportunity. And I'm sorry for changing the schedule, which was accepted by me for yesterday, but unfortunately due to some very important meeting, I'm forced to postpone, for which the management and the principal and the director are kind enough to change the schedule and gave me an excellent opportunity for sharing these thoughts to all of you. Thank you very much.
Thank you, sir. Thank you for an excellent presentation. Sir, there are a few questions from the participant side, with your permission.
Sure.
Sir, this is a question from Tapas Kumar Kuntia. Is there any other method or technology through which cargo toxicity can be reduced?
There are many methods I already mentioned. What any other method? Because there are so many methods like pegylation and increasing its hydrophilicity, etc. We can reduce the cargo toxicity to a great extent. That is one approach. And there are so many methods which can be tried experimentally in the laboratory or in the industrial scale.
Thank you, sir. This is a question from Praveen Pasala. Is there any specific reason behind clinical failure of carvedilol liposome with ketosone coated formulations?
No, it is not I don't say it is a failure, because it is not economical to carry it forward for commercialization. That is my opinion. Because if you take the carvedilol drug and its availability in the market, if you prepare a liposomal formulation and market it, the cost is going to be minimum 10 to 15 times higher compared to the conventional carvedilol dosage forms already available in the market. So for a hypertension disease like people may not be able to afford the cost, and hence certainly people may not like to use this. For example, I give you a small crude example. People may curse me. The Zydus Cadila is making diclofenac transdermal patch, which is lasting for only 24 hours. But the cost of that patch is around 45 rupees. Whereas for treating the same analgesic pain, what we are suffering from post-operative care or the rheumatoid arthritis, we can purchase 50 paise to 1 rupee diclofenac sodium tablet, and we can take it 3 times a day, which is costing me not more than 1 rupee to 2 rupees. So if I am prescribed with the drug of transdermal patch, I certainly ask the doctor, why you are giving me this drug? Spending 45 rupees a day for a transdermal patch, which also gives a 24 hours relief, why should I go for 45 rupees a day? That is the logic what is behind for not making some drugs into the nanopharmaceuticals applicable for various diseases. But for a drug, for a disease like cancer, certainly we have to use these nanopharmaceuticals because the side effects are more if you use a normal conventional dosage form. That is the reason why I feel carvedilol may not reach the market. Not only carvedilol, even the drugs that are meant for Alzheimer's disease, Parkinson's disease are not there in the market still, though they have developed so many nanopharmaceuticals for these two diseases. In spite of claiming that crossing the BBB is a very difficult task, and they have achieved this BBB, crossing the BBB at a very great extent, but that was not commercialized because of the cost effectiveness. My opinion is that.
And that question, sir, from Praveen Pasala. So kindly suggest me best nano formulation for nasal delivery.
Nasal delivery, I think these solid lipid nanoparticles or polymeric nanoparticles or even liposomes, if we can stabilize the liposomes, they are the best formulations because they can be given in the form of a liquid and it can be given in the form of drops. So I say yes, but only problem is stabilization of these liposomes for the nasal delivery. If you can do it, if you are successful, liposomes are the best formulations, I suppose.
So this is a question from P. Chandrakala. How nanotechnology studies can be, nanotoxicological studies can be done in vitro?
Frankly, I don't have ready data with me, but I have collected a lot of data. It is not handy with me. I'm not a good toxicologist, honestly. I need to take the help like people like Kumar, honestly, for these studies. I always bank with them. So Kumar is the right person to give the answer rather than me. Honestly, I have to confess.
Yeah, there are any number of cell lines available, sir, for testing.
Yeah, we need to use the different types of cell lines. That is the only approach what I feel. But specific cell lines, I don't remember exactly.
Thank you, sir. There is a small announcement. Dear participants, please raise your hand if you have any questions. You can directly talk to our speaker, sir.
Yeah.
Sir, there is another question from Praveen Pasala. May I know clinical trial status for pivotal study?
Pardon?
May I know clinical trial status for pivotal study?
Pivotal study.
Pivotal study.
Clinical trial status for pivotal study? I don't understand the question, honestly.
Praveen, would like to ask the questions, right? You can unmute and ask.
I think this is a Praveen Kumar Laddu, not my Praveen Kumar Pasala.
Okay.
I think it's a Praveen Kumar Jaldu, not Praveen Kumar Pasala.
Yeah. What is your question, please?
Okay, sir. Thank you. Question, sir. May I know clinical trial status for pivotal study?
Pivotal
study.
Pivotal study. It is depending upon the study we are going to take up. Pivotal study is always taken initially before going for a clinical trial, maybe, right, Kumar?
Yes, sir. Pivotal study is the final approval stage, phase 3 study, sir.
Yeah.
So you are going to any specific indication once you do the final trial, which you call as pivotal.
Yeah. What is the stage? It all depends on the investigator who is doing that study, in fact.
Thank you very much. I request Dr. Rajkumar Reddy sir to give vote of thanks.
So, honorable speaker of the session, Professor K.V.R. Ramamurthy sir, and all the delegates present in the session, good afternoon all. I am Ravi Kumar, Associate Professor of Sri Vishnu College of Pharmacy. I take this privilege to propose vote of thanks after this AICTE sponsored STTP session. So, firstly, I would like to express my gratitude and sincere thanks to Ramamurthy sir for accepting our invitation with a kind heart. In spite of busy schedule, you spend time for us, sir. It is always fresh and interesting to listen to our scientific talks. Today, you delivered talk on nano drug delivery system, current trends, and future perspective. It is very apt for the STTP team, sir. You have provided to all the participants a lot of updates and insights on the nano drug delivery system, which are some subtopics like you have addressed the challenges of developing nanoparticles in academic and research laboratories, and regulatory issues faced by nanoparticles during the development stage, and physicochemical characterization of nanoparticles. You mentioned some methods also like counting methods and NCable methods and many more. Biocompatibility and nanotoxicology and pharmacokinetic dynamics, scale up and reproducibility, and PAT technology, that is process analytical technology, and vastly discussed on ethical issues facing by these toxicity and safety issues facing by these nano drug delivery technologies, which are connected to regulatory issues. So it is overall a big picture on nano drug delivery systems, sir. Thanks a lot for covering the vast knowledge, huge knowledge on this nano drug delivery system. I hope all the delegates have immensely benefited by this talk, especially who are working on this area. So thank you once again, sir.
Thank you, Kumar Reddy.
I express my sincere thanks to our director, sir, Dr. Kumar V.S. Nemani, for supporting this STTP by providing most valued speakers. Thank you so much, sir. I thank you to our principal, Dr. K.S. Natara, sir, for continuous monitoring and supporting us to conduct sessions successfully. I thank all the delegates for your cooperation during the session. I'm thankful to staff of SPCP for working on and off the screen, like Dr. Babiraju and Mr. V. Ramakrishna, sir, and Tejo Manasa Madam, etc. And lastly, thanks to AICTE for sponsoring this STTP, 1 week STTP. So thank you all once again.
Thank you, Kumar.
Thanks a lot for a very, very energetic and very informative session.
So normally, though you have given me 1.5 time, always consider 45, 50 minutes is my always prefer. It is a good lecture, I always feel. Instead of giving 1.5 hours, that's why I just constrained myself to approximately 50 to 55 minutes. I hope I have done my justice what I have planned.
Yeah, I think that is the 50 minutes good time for a good effect. Most of the faculty, even for students, hardly they have 30 minutes attention.
Right. Thank you, Madhavi.
Thank you, sir. Thank you very much for accepting our invitation and being with us today. Thank you very much, sir.
Right.
Thank you for the overwhelming response from the participants. Thanks for making Day 3 event a grand success. Feedback link is provided in Zoom, YouTube channel, and in WhatsApp groups. Please do feel to mark the attendance. Wish you all a very good time. SBCP, STTP will look forward to meet you on Day 4 session. Thank you. Thank you all.
Thank you.
Bye.